I'm Priyanka Burman and welcome to my class. So today we are going to discuss one of the most important yet a little bit of confusing topic, that is spermatogenesis. So if you have already studied this topic, you know that there are many terms that sound similar to each other, and then the process, like which comes before, which comes later, and what are the number of chromosomes during which stage. So everything together might sound a little bit complicated, but worry not, because in this video I'm going to clarify all of your doubts. So let us start. Spermatogenesis. So spermato and genesis, that means the genesis of sperms or formation of sperm. So the process of formation of sperm in case of the male human beings is called spermatogenesis. Now this process, it consists of many stages. It is not a one-step process, it gradually takes place. So one by one we are going to understand what are the different steps involved in this process. So here you see the first cell which is actually involved in this process is the spermatogonia. So spermatogonia is the cell with which the process will begin. Now, what is the number of chromosome in spermatogonia, or what kind of cell is the spermatogonia? It is having twice n chromosome. That means it is a diploid cell. It has 46 number of chromosomes for human beings. So this is the spermatogonia. It has 46 number of chromosomes. Now, this spermatogonia, it is going to undergo mitotic division. So you can recall mitosis, right? Mitosis. So what happens in mitosis is that the cell will start to replicate. It forms similar cells of having similar chromosome number. So the diploid cell is going to produce another diploid cell which will be similar. So this cell, what actually they are doing then? In simple language we can say they are actually multiplying the number of cells. So this spermatogonia, it will undergo mitosis and then it will become multiple in number. Now these out of all the multiple cells, few cells, remember not all the cells but some cells will undergo the process further. Not all the cells, few cells. So those few cells which will undergo the process further are called the primary spermatocyte. So the primary spermatocyte is a result of what? Mitosis of spermatogonia. So that means the number of chromosome is again going to be diploid. That is 46 chromosomes are present in primary spermatocyte. So now you see, we are considering this primary spermatocyte and we will see what happens further. So now this primary spermatocyte will undergo the meiosis 1 process. That is, it will undergo reductional division. This is the moment where the number of chromosomes will become half. It was 46 chromosome in one cell. So now it will become 2 cells, but each cell will contain 23 numbers of chromosome. It will become haploid. Yes, so secondary spermatocyte is the cell where the reductional division will happen and it will convert into a haploid cell. Then the secondary spermatocyte will complete the process of meiosis. It will again undergo the rest of the meiosis, that is meiosis 2 or second meiotic division, and it will form the final cell. These final cells are called as the spermatids. So this spermatid will again be haploid, that is 23 number of chromosome. Now this spermatid, as you can see, they are still very much similar looking to the earlier cells. They are again round in structure. They do not look like the sperm yet. So that means what? There is something more. We need to do something more in order for the sperm to be formed. So what we will do? The process is called differentiation. So the spermatid will undergo differentiation, which is actually morphological, we can say modification in order to make it more motile, so that it can move better, so that it can reach its destination, that is the female body, the near the egg of the female body. So this differentiation will lead to the modified spermatids, which are called as spermatozoa. So this time the spermatids got modified, and now they have a head and a tail. You can see very easily they have a head and a tail. The head became smaller. There is a tail which is like a flagella, flagellated cell. It helps in movement. So these spermatozoa are now similar to what we usually draw for the sperm. But now you might think that okay, then fine, the process is over. But no, the sperms are still not ready. So now what will happen? These cells, the spermatozoa, will now get embedded. That means what? They will get entered into what? The Sertoli cells. Suppose this is the sperm, this is the Sertoli cell. The head portion will get embedded into the Sertoli cells. So what are the Sertoli cells? They are the nourishing cells. They will provide nutrition. They will provide nourishment. Now this nourishment is given to the spermatozoa, and then finally they will be ready to leave the organ, or they will be free to move out of the organ to carry out the function. So you see, now we can say that finally the whole process of spermatogenesis is over. Now there are certain terminologies which is used for the different stages of this whole process. Now we are going to learn the terminologies. So first of all, what was happened from the spermatogonia? We got primary spermatocyte, and then we got secondary spermatocyte. So here what was happening? Spermatocyte was being produced. There was genesis, production of spermatocytes, primary and secondary spermatocyte. So these area, you see from here to here, this process we can use under one terminology, that is spermatocytogenesis. So this is another terminology that we got, spermatocytogenesis. Again, for the next phase, if we see, where the spermatid differentiates into spermatozoa, that is the process of modification of the sperm from spermatid to spermatozoa is called what? Spermiogenesis. So again there is production of sperm, there is genesis. So this is spermiogenesis. Now there is another similar term to spermiogenesis, that is spermiation. So now you might be confused between spermiogenesis and spermiation. So remember, genesis, there must be production. So that will be involved with this differentiation stage. And spermiation, it is simply a process in which these cells used to get embedded in the Sertoli cell. They got nourished and now they are free to leave. So this process, where the cells or the spermatozoa become free to leave, that is called spermiation. So we got three terms: spermatocytogenesis, then we got spermiogenesis, and then we got spermiation. All these three together, we complete the process of spermatogenesis. And then if we talk about, questions that may be a little bit uncommon for you, then let me tell you, first of all, what is important is that which cells are haploid and which cells are diploid. That you have to, understand very well. So here you can easily understand it, I believe, because first mitosis was there. So during mitosis, definitely there is no change. And where change occurred? During the first meiotic division that took place after primary spermatocyte. So here in these two stages, we have diploid cells and the rest are haploid cells. Next thing that they can ask is that how many spermatozoa are formed from one primary spermatocyte. So if we have one primary primary spermatocyte, then how many spermatozoa can we get? So here you see we considered one here. So from one primary spermatocyte we get 4 spermatozoa. One primary spermatocyte we get 4 spermatozoa. Similarly, for secondary spermatocyte we get, you see, from one secondary spermatocyte if we see, we get 2 spermatozoa. Okay, so primary 4 and from secondary we get 2. So if the question comes that suppose we have n number of primary spermatocyte, then how many spermatozoa are produced? So what you will do, you will simply multiply n by 4 and your answer will be there. Similarly, if the question comes for secondary spermatocyte, you will simply multiply it by 2. Understood? So these are the 2 questions that sometimes puzzles the students. Then rest, these terminologies are important, and like I said, the stages, number of chromosome is important, and then finally your whole concept is cleared. So I believe with this you have understood the whole process of spermatogenesis. Thank you.